Thank you very much for the kind introduction, and a very good morning to all of you. Before I start, I would just like to give my special thanks to Philippe, who has given me this wonderful opportunity to be here today, and it is an honor. So I'll be giving you an overview of what I have been working on the past few years on Chikungunya at SINE, with a special focus on human immunology. Now, I believe that Chikungunya is nothing new to the French. I'm sure you probably know more about Chikungunya than me. But for the benefit of those who are not familiar with this strange disease, I'll just one slide that captures all the important pointers. So at the end of this talk, you should know everything about Chikungunya from this slide. So it is an alpha virus, which is transmitted by the Aedes mosquito. And during late 2005, it has suddenly re-emerged in the Indian Ocean islands that has spread to several countries in Southeast Asia, including Singapore. Now, the typical acute infection probably lasts about a little more than a week, with high fever and painful arthralgia. So earlier on, my group actually did some electron microscopy, and we found that in terms of size, it is pretty similar to other alpha viruses of about 0.1 micron. And in terms of its genome, it is a positive RNA virus of about 11.8 kilobase pair. Now, this is a slide that was obtained from Ann Powers from the US CDC. She has worked for more than 20 years in alpha viruses. And before that, she documented the distribution of chikungunya around the world, and you could see from here that it is mainly in countries in Africa, some parts in India, and also Southeast Asia. But what happened after 2005 to 2006? Now, this is not updated anymore, but you could see that there are several countries who have reported cases of chikungunya, now including Singapore. Now, clearly, the changing disease patterns and the spread of the Aedes vector in several temperate countries introduced new risks of epidemics in countries with a naive population. So Singapore was also not spared. Now, there have been several hypotheses that suggested the re-emergence. So many people have been speculating about climate change, the absence of herd immunity. But I'll just like to bring to your attention on some studies that were done in a Scott Weaver's group in the US. And they have found that there was this specific mutation in the E1 glycoprotein with the mutation of the alanine residue to a valine residue at position 226 actually increased the viral replication in the Aedes albopictus. And incidentally, in most of the Indian Ocean islands, the main vector was also Aedes albopictus. So this potentially could have caused the sudden outbreaks. And just early this year, they have continued with a follow-up study, and they have found that together with another mutation in the E2 glycoprotein, the L residue to a Q residue will further increase the dissemination. So you need more than one mutation for such increase replication and virus replication and dissemination in the albopictus. So just try to recognize the two little mosquitoes down there. And I just want to mention that in Singapore we have both mosquitoes, so please be careful the next time when you visit. Okay, so chikungunya in Singapore. So I mentioned we have a few waves of outbreaks since, in fact, late 2006 we have been sporadic imported cases. But I'll just like to highlight that since 2008 to 2009, we had a few outbreaks, and both of the different vectors are also involved. And for the rest of the study of this morning, what you will hear are the studies that were performed on the patient cohorts from these two outbreaks. So the focus in SINE, as I mentioned, was very much on human immunology. So we wanted to establish, to get more information on the immune responses that are mounted against this virus infection, and how could we use the information to develop new immune-based and control strategies. Now, after the first outbreak in early 2008, we made use of the Luminex platform that was just established then to identify possible pro-inflammatory immune mediators during the acute phase of the infection. Now, from here, we were able to identify various factors that were found to be involved in causing some of these acute symptoms, namely We have interferon alpha, IL-6, MCP1, and IP10. But what is more interesting and more important are the factors that contribute to the chronic symptoms of the disease. So with the subsequent outbreaks, with a larger cohort of patients, we were able to follow up longitudinally and collect the samples up to the chronic phase. And from here, we found markers that are associated with the chronic joint pains. Now just have a look at this slide. Now we found that for patients, we still had the persistent joint pain during the chronic phase, still had higher levels of IL-6 and GM-CSF. And I'd just like to highlight that these two factors were also pretty much associated with patients in rheumatoid arthritis. So this is just one of the earlier reports. And more recently, working together with Suresh Mahalingam from Griffin University in Australia and Pierre Hooks from the CEA, so we were able to extend this study further and found that both rankle and OPG had different production profiles in the chikungunya patients, so thereby suggesting a possible role of the osteoblasts in contributing the atralgia from the CHIKV infection. Now we could also see that the levels of OPG and rankle also associated with the viral loads. Now I'm sure if you could discuss with Pierre more later, if you are interested, she would tell you more about some of the studies that's done in the osteo class. Okay, so we have seen the early infection, and we made use of some of the immune mediators profile. And there's another point which I would like to highlight, which is the role of the immunoglobulins. So, are they protective? What do we see? So in order to quantify the antibody responses, we made use of the same cohort of patients to further define the antibody profile. And we could see from here that starting from the acute phase to the chronic phase, the profiles are pretty much well defined. And as expected, you could see that the IgG starts to produce at around the early convalescent phase, and then they rise that remain fairly stable after the chronic phase. And you could see that although the IgM peaked earlier, they also started to decline earlier. And as we all know, that the IgG has various isotypes, each with a specific property and also biological functions against the different antigens. So we went on to isotype these antibodies, and when we did that, we found that these chikungunya antibodies were exclusively of the IgG3 isotype. And what is more interesting was that we observed in our cohort of patients that about half of them actually had higher levels of the IgG3 during the early convalescent phase, while the other half of the patient, they had IgG3 levels that are still fairly low. So therefore, we segregated this group into what we call the early IgG3 group and the late IgG3 group, because towards the end, the levels were pretty much similar. So with this, we went on to identify if these CHIKV antibodies had any protective capacity. So the next thing was in vitro neutralization assays. So we then incubated a virus with the patient's antibodies, and we then infected a susceptible cell line 293T with this complex. And we made use of the Cellomics high content screen to measure the readout. And you could see from here, indeed, these CHIKV antibodies from these patients could actually neutralize the virus in vitro. And in fact, the early induction of these anti-CHIKV antibodies actually shows stronger neutralizing activity against the virus in vitro. So, but are these antibodies neutralizing antibodies of the IgG3, since they are supposedly clearly exclusively of the IgG3 isotype? So the next thing was we tried to deplete the IgG3 fraction of this patient's antibodies in this depletion assay, and then we measured the ELISA titer, and you could see from here, with the depletion of the IgG3 antibodies, it translates to a reduced neutralizing capacity of these antibodies. thereby confirming that this CHIKV specific IgG3 isotype is important in the neutralizing activity. Now, putting some of this in vitro data together with the clinical findings, we found that in the patient cohort, the early IgG3 groups actually had higher levels of viral load during the acute phase of about 4 days, compared to those group of patients that are late IgG3, but they had a lower level of viral load. And these patients also had a higher incidence of what we call severe disease. Now, in Singapore, the spectrum of chikungunya was fairly narrow, so the clinicians actually classify severe disease in terms of fever, the temperature, the pulse rate, and also the platelet count. So these patients, early IgG3, had higher viral load, more severe disease during this stage. But with the production of the early IgG3, they were able to control and clear the virus at a faster rate compared to the other group. And this group of patients fully recovered towards the chronic phase, compared to the patients with the late IgG3, less viral load, less severe disease during the acute phase, but 30% of these patients went on to continue to have the persistent arthralgia. So with all these, we found the neutralizing antibodies, the IgG3, and we wanted to identify which are the surface antigens that are targets of these neutralizing antibodies. So we assess the surface antigens, which are mainly the structural proteins, and in order to do that, we assess each patient's plasma through Western blot, and when we scan all the blots of these patients, we found that the E2 is the immunodominant viral protein upon CHIKV infection. And with that, we have identified the E2 that was the main viral protein during this disease phase of median 10 days. We wanted to identify the specific epitopes that are responsible. So with that, we made use of an overlapping library, of a library of overlapping peptides, and we found that the patient's plasma actually specifically detected the first 2 peptides of the E2. And with the available crystal structure of the E2 glycoprotein by Philippe Kourilsky at the Pasteur Institute, we were then able to locate the location of this specific epitope, which we now call E2EP3. And you could see from here that it is actually at the surface and is very exposed comparatively. So with this, with the sequence, we wanted to identify the key amino acid residues that are important for binding and for recognition. So we made use of a series of alanine mutated amino acids, and we did the alanine scan. And we found that particularly there were 3 amino acids that were important. And where are they located? You could see very clearly. It's down here. Now, I just want to highlight that the amino acid number 3 was not resolved in the structure, so we were not able to locate it. But with this, we wanted to confirm the importance of E2EP3 in the neutralizing capacity. So what we did was, we incubated the free E2EP3 peptide with the plasma antibodies, and we did the in vitro neutralization assay. And you could see from here that the addition of soluble E2EP3 peptide to the plasma was able to partially abrogate the neutralization activity. And in order to go a step further, we also generated the alanine mutated peptides, and we tried to deplete the E2EP3 specific portion from this patient's plasma. And you could see from here that E2EP3 was able to deplete most of the antibodies, while the alanine mutated peptides were not able to deplete them fully. And this, of course, translates to a reduced capacity of neutralization, as you could see from here. So with this, we confirm that the depletion of the E2EP3 specific IgG3 antibodies abrogates the neutralizing effects. So, but does E2EP3, could it be detected by the different patient cohort? So we extended this screen from the other patients in Singapore, and working together with colleagues from the University of Malaya in Malaysia, we also managed to screen some of these patients. And from here, we found that the E2EP3 is a common detection serology marker for early CHIKV infections. Now, I just want to mention that for all these patient samples here are all within 14 days post-infection. Now, we all know that the non-human primate model is also frequently used for viruses. And working together with Pierre Hoeks from the CEA, we were able to obtain some of the materials of the CHIKV-infected macaques. And you could see from here, once again, plasma from this CHIKV-infected macaques was also able to detect, be detected to recognize E2EP3. So making this a very versatile region. So what next? We have identified the key neutralizing capacity of the E2EP3. They are able to be picked up against various cohorts. So the next thing was, could E2EP3 function as a possible vaccine candidate? So with that, we covalently link E2EP3 to a KLH, and this was done in collaboration with Karl-Heinz Wismuller from the EMC in Germany, and we immunized the C57 Black 6 mice with this so-called E2EP3 vaccine. And interestingly, we could see that it does induce a very, very good level of antibodies in these vaccinated animals. And making use of this reported wild-type adult CHIKV model that was reported by Andreas Serbier from Australia, they were able to mimic the human infection in terms of viremia and joint inflammation. We challenged these animals with the virus. So you could see from here that the vaccinated animals were able to have a lower levels of viremia, and serum that was obtained from this immunized and challenged animals were also able to have a fairly reasonable level of neutralizing capacity. Furthermore, these animals were also somewhat protected with a reduced viremia. They also had a reduced joint inflammation. Now, so moving on from here, it is important to identify if there are other potential epitopes that perhaps may be less dominant than E2EP3, but nonetheless important for neutralization, and this could be used for further vaccine designs and also seroepidemiological studies. So with the availability of the longitudinal samples of up to 2 years, we then detected some of these regions and also mapped them to the specific antigens, and we found that the NSP3, capsid, even the E3, and other regions of the E2 are also important. So from here, what you have seen here today was with the various approaches, we have found markers of infection, pathogenesis, and also protection. And this could actually form the basis for future translational studies. So before I end, I'll just like to give the acknowledgement to the various members, especially the hospitals and the clinicians, for providing the patient cohort. And particularly, I'd just like to highlight 2 colleagues from SIGN, Laurent Renier and Olaf Roschke, who actually have been my most critical colleagues since I've started, and with their constant criticisms, it has actually pushed me to keep trying to do better and better. And also to Pierre Hoeks, my close collaborator from CEA and also a dear friend, and from Gilles, who has kindly provided us with some of the synovial sites for our study in Rankle and OPG. And last but not least, to Philippe, who has been always challenging me and teasing me to start this Chikungunya program when I first joined SIGN. So thank you for your attention, and I will be happy to take on any questions.